Use of Colorimeter. This video will demonstrate how to determine the concentration of a sample solution by using a colorimeter. Colorimeter is a device to measure the absorbance of a solution at a particular wavelength of light. The data can be used to determine the concentration of a solution. In this video, the absorbances of several standard copper 2 sulfate solutions of different concentrations will be measured first. From the data, we can draw a calibration graph of absorbances against concentrations. Then, the absorbance of copper 2 sulfate solution with unknown concentration is measured, and the concentration of the unknown solution can be determined. Before the measurement, we need to select an appropriate filter. Insert the chosen filter into the colorimeter. To obtain an accurate measurement, the color of the chosen filter must be the complementary color of the sample solution. For example, copper 2 sulfate solution is blue in color. The complementary color is red. So a red filter is chosen. First, set the absorbance reading of the calorimeter to 0. Rinse the test tube for containing the sample solution with deionized water. Fill the test tube with deionized water. Fill the test tube to about 2/3 of its volume. Put the test tube with deionized water into the colorimeter. Add marks on the test tube and the colorimeter to ensure the test tube is placed in the same direction in each measurement. The direction of the test tube will affect the accuracy of the measurements. Place the test tube cover. Adjust the colorimeter until the absorbance reading is 0. Take the test tube out of the colorimeter. And discard the deionized water inside the test tube. Rinse the test tube with a standard solution twice. To ensure the accuracy of the calibration graph, start measuring the standard solutions from the lowest concentration. Measure the standard solutions from low concentration to high concentration. Add an appropriate amount of the standard solution into the test tube. Gently wipe away the water on the test tube surface with tissue paper. Put the test tube into the colorimeter. Make sure the direction of the test tube is correct. Cover the test tube. Record the absorbance reading for the standard solution. Take the test tube out of the colorimeter and discard the content inside the tube. Select the standard solution of the second lowest concentration. Rinse the test tube with the standard solution twice. Add the appropriate amount of standard solution into the test tube. Record the absorbance reading. Measure and record the absorbances of the 5 standard solutions of different concentrations in correct order. Plot a calibration graph with the results. The calibration graph gives a straight line. It means that the absorbances are directly proportional to the concentrations of the solutions. Measure the absorbance of the copper 2 sulfate sample solution with unknown concentration. Take the test tube out from the calorimeter and discard the content in the tube. Rinse the test tube with deionized water 2 to 3 times. And then rinse the test tube with the sample solution twice. Add the appropriate amount of the sample solution into the test tube. Put the test tube with sample solution into the calorimeter for measurement. Make sure the direction of the test tube is correct. Record the absorbance reading. In this measurement, the absorbance of the unknown sample solution is 0.50. From the calibration graph, the concentration of this unknown sample solution is determined to be 0.071 molar. After measurement, take the test tube out of the calorimeter. Rinse the test tube with deionized water. Take the filter out of the calorimeter and put it back into the container. Put back the sample cover.